Hello everyone, good morning all of you. Today episode 4 series of national talk on the topic science of variants, vaccine testing and their management. This national talk series organized by the Fit India Campaign Committee and Fit India Club of the Manipur University as a part of the Fit India movement during COVID-19 second wave pandemic. Well, in this occasion, let me welcome each one of you present over here. First of all, I would like to extend my sincere welcome to the panelists. Professor L. Shanjukumar Singh, ISOD Biotechnology, Manipur University. I welcome you, sir, as a speaker for this session. And I thank you, sir, for your kind accepted our invitation to be a presenter and speaker for this session. Thank you so much, sir. I respected Dr. C.H. Ibol Singh, Associate Professor and Head, Department of Library and Library and Science, Manipur University, as moderator. I welcome you, sir. And respected Dr. C.I.S. Ras Ketan Singh, Associate Professor, Department of Manipur University. I welcome you, sir, as a co-moderator for this session. And especially, I would like to a warm welcome to all the participants, learned member, faculty member, and non-teaching staff, students, everyone present over here. I welcome you. And I would like to welcome our media partner, our Mami Devi also. I would like to welcome for this session. And now overall, okay, so everything set now. So well, I would like to request again for today co-moderator Dr. C.S. Ras Ketan Singh, Associate Professor, Department of Manipuri. Sir, please have introduced today's speaker, Professor L. Shanjukumar Singh. Please, sir.
Thank you, Dr. Santosh. A very good morning, everyone. Let me highlight the biodata of Professor L. Shanjukumar Singh, today's speaker. Professor Lisam Shanjukumar Singh is presently the head of Cancer and Molecular Division Department of Biotechnology, Manipur University. He got his PhD degree from NEHU, Shillong in 1998, and from 1999 till 2009, he carried out his postdoctoral in USA. His research area include cancer biology, cell and molecular biology, anti-cancer medicine plants, HIV, epidemiology, etc. Some of the awards he received are the Cleveland Clinic Innovation Award for development of a method for the inhibition of metastasis in 2002. Achievement for Excellence in Cancer Research, Indiana University Cancer Center in 2006. He has more than 38 publications in the 3 patents to his credit. Thank you.
Thank you, sir. So, well, we have to proceed our program. So I'd like to request today moderator, Dr. C.S. Ibol Singh, to have the session. And I would like to hand over the session to the moderator. Thank you, sir.
A very good morning, everyone. This of the webinar conducted by Fit India Campaign, Manipur University. In this very fine morning today, we have an invited speaker with us. We don't find a better speaker today other than Professor Shanjukumar Singh. whose brief biodata has already been highlighted by Dr. Rajketan Singh. So without wasting much of your valuable time, let us try to have a glimpse of the deliberations to be made by Professor Shanjukumar Singh. Today we are very much talking about different variants of COVID-19. So many vaccines have been developed across the world. Same thing is happening in the case of India too. So many testing have already been conducted in this very part of Indian subcontinent too. And let us see the signs of variants of this COVID-19 and how different developed vaccines have been utilized to control the pandemic and the way how it managed. So Professor Shanjukumar is the right person to speak on the theme. So without wasting much of your valuable time now, I, on behalf of the Fit India Campaign Manipur University, would like to invite Professor Shanjukumar Singh to take his valuable time. Please. Over to Professor Shanjukumar Singh.
Okay, thank you, everybody. Am I audible to everybody? Hello.
Yes, yes, sir. Yes, sir.
Okay.
Yes, it's audible.
First of all, I thank the Fit India Campaign Committee and the Fit India Club Manipur University for inviting me for this talk. And I thank all of you for participating in this talk. The title of my talk is COVID-19, Science of Variant Vaccine Testing and their Management. So before starting the main topic, let me start with some introduction so that it will be easier to understand the main topic. So I'll start with some basic of some micromolecules that are important in the living cells in life. So there's nucleic acid and a protein. So in the nucleic acid, there are DNA and RNA. DNA is in most of the living cell, DNA is the genetic material. It is double-stranded, and RNA is single-stranded. So from DNA, RNA are synthesized through transcription, a process called transcription. Then RNA synthesize protein through a process called translation. So DNA is just like a hard disk, and it synthesizes RNA. When it synthesizes RNA, the sequence of the DNA is directly copied to the RNA. There's one base different, of course. Then from RNA, whatever the sequence, the nucleic acid sequence, it translates into protein. So in short, DNA tells what type of protein, which protein will be synthesized. So and the protein is the functional molecules, is structural and functional molecule of the living system. Most of the enzyme, most of the protein, for example, our nail or everything, it is, and the activity of cell, movement of cell and everything is done by the protein. Except few virus, their genetic material is RNA. In this case, the COVID-19 SARS-CoV-2, the genetic material is RNA. It's not the DNA. So RNA can be converted to DNA through a process called reverse transcription, catalyzed by the enzyme reverse transcriptase. So in a very simple way, that whatever sequence of the DNA is again transcribed into RNA, and from that RNA sequence, protein are synthesized. So when there is a mutation, in the DNA, then there is a change in the amino acid of the protein. So when there is a change in the amino acid in the protein structure or function will change. For example, there is a point mutation in the DNA, then the protein, one amino acid difference will make some difference in the protein property. For example, say let's consider a protein called a sentence, this is a boy, but there is a one letter mistake, it is changed to T, then its meaning is there is a toy. So the meaning is quite different. Similarly, when there is a mutation in the gene, the protein which is synthesized by that protein, the property, the structure changes. So let's come to the virus. This virus, COVID-19 virus, is a very simple. Most of the virus are simple, and the weakest living particle, they are not called as cell. They are called as particle because they are in between living and non-living. That's why they are called as particle. So they have this genetic material inside the capsule. Then some protein are protruted from the membranes, from the capsule. Then this protein does a lot of function for the virus. So let's see what the protein does. In this case, I'd like to give attention to this particular protein, spike glycoprotein S. This is also known as specifically S protein. And this figure 2 shows the cell of human being, very organized higher cell. So if we compare these two, this human being cell, they are quite complex. They have a lot of inside the cell. They have a lot of organelles, and the compartments, they have different functions. So this living human cell can do a lot of function independently. For example, they can divide independently without any cells help. But in case of the virus cannot replicate by themselves because they don't have the machinery for the replication. So virus always needs some host cell. So in this case, in this COVID-19 virus, the human cell is used for its replication. So I'm showing one green structure there. If we consider this a receptor, in the human cell, this spike protein will bind to this human receptor. This human receptor is known as ACE2. So this virus genome, that is RNA, is very short. I mean, it's very small, only 30 kb length. You compare with the human, it is about 6.25 gigabase pair. So this virus, COVID-19 virus genome, have an instruction to synthesize its own protein. This one is the spike protein. Then is some other endonuclease and helicase, like that. And these proteins are necessary for their replication. And this spike protein is the protein which outside the virus structure. They're protruted outside the virus structure. So this one is important. The three-dimensional structure of this S protein is shown here in this picture. So this is suppose let's consider this is the virus, and the red structure protruded from the virus is the protein, spike protein or protein S, and it binds to the human receptor ACE2. And this human receptor ACE2 is not the natural binding site for the spike protein of the virus. It is not a natural, but somehow this virus gained the ability to bind to this human receptor. This human receptor, that ACE, it has one protein that it binds. So it's just like this virus forcefully bind to the human receptor and used to enter the human cell. So spike protein or S protein is very important. So let's talk about the COVID, origin of COVID, little bit, not much. At first it was believed, or it was said. I don't want to say it exactly what believed, but it was said that this virus originated from bat. And after some months, then they said again it is originated from pangolin. And there's another lot of theory is there. First it was in the bat, then it goes to the pangolin. Or some other theory also there that this is some kind of bio warfare virus. Means bio warfare means they manipulate some virus and make it very, very dangerous, infectious. Then they used to the enemy people. Some people believe like this, and some again believe that it just escaped from the research lab. So it's in Wuhan, where this virus infection was started in the first place. There's a virology lab. So they do a lot of research. They handle very dangerous microorganisms. So people believe that it is escaped from there. Or some others are saying that combining with the military and the civil research lab. So there's a lot of theories going on. And finally, then the first theory is that this virus naturally jump from animal to human. If it is true, then it's not the first case that an animal virus jump to the human. For example, say HIV. Most of you might be knowing the HIV it jump from monkey to human. And the second theory is that it's a man-made, as I mentioned, is like escape from the laboratory, biology lab, or some biotourism lab for using it for bio warfare. So whether it's coming naturally from the animal or is the man-made, now it attacking the human being, which is believed to be supreme on earth, the strongest living thing on earth, the human, the powerful living thing on earth is attacking the human. So there is a lot of politics is going on. The USA and other countries, led by US, they said we need to find out the origin of this virus because we suspect something. It's not from the animal. It's not natural jump from the animal. That's what they believe. The USA and other countries believed. And China, they don't want to investigate. They said no need to find out. It's not necessary now. People are dying. We should concentrate on the people dying for their health care. It's not time now to find out what is the origin of this virus. At the same time, they are hiding. They are hiding. They are not cooperating with other country to find out the origin, because naturally this virus is come from their place. And at the same time, this World Health Organization, they don't know what to do. They are just in between, balancing themselves to which side they have to go, because they have a lot of China also funding a lot of money on World Health Organization too. And very interestingly, just yesterday, last night, I found very interesting news is that some scientists, two scientists from British, they have proved that it is not the virus, the COVID-19 virus, SARS-CoV-2 virus is not a natural virus that jumped to human. It is a manufactured by genetic engineering. It's going to be published very soon in a journal, Biophysics Research. What they said, what the Chinese, I'm very sorry. What this British two professors are claiming is that this human receptor which is not the natural receptor of the virus protein S, it has negative charge. Then spike protein has 4 positive charge amino acid in a row. So what these scientists are showing is that it is very unlikely to have positive charged amino acid in a row 4 times. Even 3 positive charged amino acid in a row is very, very, very rare in nature. And now 4 is this impossible. So it is somebody have engineered, is somebody manufactured purposely to kill people, to infect people. That is these 2 British scientists are claiming. Anyway, so let's compare the size of this virus is very small, and if you compare to the bacteria is much bigger. Now again, if you compare with the human red blood cell, it's the comparison size. So like this virus, compared to the human cell, it is around 50 times smaller than human red blood cell. Again, this human red blood cell is the smallest in the human cell. Among the human cell, red blood cell are smallest. Usually other cell, their size is around even up to 17 micrometer. So this is just a structure how the human cell are infected with the virus. Okay, let's come to the some fact. Now the fact is this weakest living virus particle, as I mentioned, that is they cannot replicate themselves. So it is very weak. This weakest living particle has killed millions of people in a very short time. And again, this smallest living virus particle has put billions of people under house arrest. No authority has done so far, but this weakest, this smallest virus has done. And now this simplest, who has nothing but some protein and the nucleic acid particle, has become the highest authority in the world. Now let's come to the life cycle of this virus. As I mentioned, that this is a SARS-CoV-2 virus, and it is the red one, or to get from the virus is the S protein or the spike protein, and it binds to the, as I mentioned earlier, it binds to the ACE2 receptor of human cell. And another protein, it helps for binding. And this, as I mentioned earlier, it is not the natural. This virus are not supposed to bind to this receptor. If you look at the bat and the animal virus, this same virus, I mean, that found in the animal, they have this S protein also, but they cannot bind to this receptor. Somehow, some as I mentioned earlier, is somebody have engineered. So it has gained the ability to bind to the human receptor. So it's just like somebody has, some thief has broken the door. This is the foreign, the virus is the foreign particle. So this virus is just like a thief and break the door through this human receptor and get inside the human cell. And once get inside the human cell, then it releases the genetic material, RNA. Then this RNA will be replicated after synthesizing some its own protein using the human cell protein synthesis machinery. The virus hijacked the human cell machinery and it synthesized its own protein. Then and again it replicate means make a lot of copy of this virus RNA. So in the cell now, the virus has protein. It has synthesized a lot of its own protein. It has replicated a lot of RNA copies. Then they pack all this together and make new virus and release out of the cell. In this way, in a single human cell, thousands of virus particles can be released, and finally, the cell will die. That human cell will die. So it's just, so what I want to say is that it's just like the virus is breaking our door. So let's talk about the variant. Variant is because of the mutation in the virus RNA. There are so many mutations so far reported, and it's continuing till now. New mutations coming up, but so far there are very important top 11 important mutations that are prevalent in USA in the world, and it's listed here. So top 1 is prevalent in US at the same time all over the world. Mutation is a natural process for all living organism, whether the DNA or RNA. Mutation is a natural process. Virus involved through mutation. And luckily, this virus has some troop reading activity, means when they replicate, mutation is less. The mutation is like, say, 1 to 2 new mutation every month. It is compared to the HIV virus, is slower. HIV virus mutation is very high. And there are some mutations that E484K and L452R mutation. Sometimes they just appear in the United States, and after some time, then they stop growing. So because of the this mutation, accumulation of mutation in the virus can be studied. This mutation can be studied. This mutation can be compared with the original sequence of the virus found in Wuhan. They compare with the original sequence of the virus, which was first found in Wuhan, and other viruses which are found in other part of the globe. When they collect all the sequence and they compare, some sequence become a group. Some virus, they disappear. For example, down here, they have some variant, and this variant stop. So this sequence was analyzed in 2021, February 1, February 15, March 1, March 15. So with time, how much the variant, how many types of variant are appearing? So among the variant, there are some interesting means not in a good sense, but to look upon about this mutant. The first one is B117. It was detected in United Kingdom in late December 2020. And another one, B2525, is also from United Kingdom, simultaneously from Nigeria in December 2000. Then another one is B1526 is from New York, United States. You remember how New York was bad at the first wave, and it was detected in November 2020. And similarly another virus. And now in India, there are 4 variants: B1617, B1617.1, and B16172, B16173. And another variant that is known as P2, it was first detected in Brazil. These are the variant that spreading throughout the world. So there are some variants who are concerned in India. Why they are concerned? Because they are spreading in India, and they have gained some characteristics. So we look into that. So the variant of concern in India, the first is the B117 UK variant. Then B1351 is a South African variant. Then Brazilian variant, then another California variant. Then there's another one. It's known as double mutation. Indian variant with double mutation. It is said that it is currently responsible for the second wave in India. But there is a doubt, there is a debate, because the mutation found in this double mutation variant of India is already detected in some part of the world. In USA, it has been detected, in Brazil and South Africa. So it is, some people are arguing that this is not a new variant. It's not an India variant. So let's see how this the most dangerous variant of UK, how it is spread through the world. In the beginning, on first wave, this UK variant was spread throughout the world, including India. Then another is South African variant. It also spread throughout the world, but it was not found in India. And another P1 Brazilian variant also spread to other part of India, but not in India. This we can believe, but I really don't know. At that time, India had started sequencing. So I really don't know. If it is not, if India variant are not sequenced, then how do we know? Okay, now some virus, this COVID-19 virus from India sample has been sequenced. This sequence was reported in 2021 March 25, and that sequence only around 771 total. Is very less, and we found that the UK variant is the maximum, around 97% is the UK variant. And the South Africa variant is very less. And Brazilian variant is very less. And where is the India double mutation variant? It's not there. But this data might have changed a lot by now, if they have the India government has done the enough sequencing, this picture, this number will have changed a lot, I believe. So this UK variant, it has around 23 mutations. When you compare with the original Wuhan variant. And there's 8 mutations at the spike protein. Spike protein, if you remember, is the protein that binds to the human cell. So let's see that some important concern about this variant. The UK variant, between 40 to 70% more infectious than the other variant, is dangerous. It increased the death risk by about 60%. But some vaccine may work against it. Then Brazil variant, then South African variant. This South African variant also, it may affect our immune system. We'll come to our immune system later. This India variant is already that mutation already found. I just mentioned some times back. It's already found. So why why they call it India variant and double mutation? Some people are still arguing, saying that it's not called as Indian variant double mutation. So we have talked again. We have talked about this S14, the spike protein. It's very important in all aspects. Throughout the talk is about the S14. This is very important. So when there is a mutation in the S14, it's likely that it increases the affinity of binding to the human receptor. Higher affinity means, increased affinity means it can bind to the human receptor very strongly. that means it can easily enter to the human cell. So it becomes very much infectious. So if you look with a closer to the structure of this S protein, it looks like this, and it have some lobes. It's that some lobes are shown with a red ring. And look at this, the right side diagram. That if as long as these lobes open, now from this figure, this one it has open and more open now. As much as it's open, it binds to the human receptor very tightly. So there is a mutation, accumulation of mutation in this protein, then it's likely that it'll open the structure will change. I said when in the first 2 or 3 slides, I already mentioned that when there is a mutation in the protein structure will change. The activity will change. Similarly, here also, when there is a mutation in this S protein, it changes the structure, it changes the activity. As long as the mutation accumulate in this protein. For example, the UK variant has 8 mutations only in this S protein, spike protein. So there is such a changes, and it becomes more open and it more fit to bind to the human receptor very tightly. So why this Baryon genomic sequencing of this virus is important? It's not just for the scientific interest. It is important for the public health. The reason is, if we know the variant, which type of variant and what are these characteristics, we'll understand the ability of the variant to spread more quickly in people. And we can predict that the ability to cause whether it's a severe disease. Now in the second wave, people are saying that this is the severity of the disease is too much and is spreading quickly. It's because of the mutation. Then ability to evade the detection by specific diagnostic tests like RT-PCR is very interesting, because some doctor, even in Manipur, they have said that they don't trust the RT-PCR result. Not only in Manipur, outside the Manipur also. That's the reason, because when you do the RT-PCR, there's a primer. Primer has to sit to a sequence, to a particular sequence of the viral RNA. The site where this primer is going to sit, if it is mutated, the primer will not sit. Then the test will become negative. Although the virus is there, but it will show negative, false negative. So there are many things that we can understand and apply for the health for the public health if we know that variant. I had some discussion in the TV, ISTV, Impact TV. Some people start talking about the the sequencing of this COVID virus circulating in Manipur. The first question is that is it better to is it necessary to know? In my mind, I think it's better to know the variant. However, forget about Manipur. In whole India, in March 2021, only some sequencing has done. So even if it's important in Manipur to know the variant of sequence, but who is going to do? Compared to the other state, our state is very small. I don't think that government would permit or give fund for sequencing. So it's in my mind, I think instead of sequencing the virus genome, it's better to use the resource for more testing by RT-PCR. I will come later on why I said by RT-PCR. But is it possible to do the sequencing of this genomic in Manipur? Yes, I think it's possible. Some people, some in the TV discussion, they mentioned that for sequencing, that BCL biosafety cabinet of level 4 is necessary. I don't think so. I don't think so. It's necessary biosafety level 4 for COVID-19 virus genome sequencing. The reason is we are doing RT-PCR. When we do RT-PCR, we take the sample and isolate the RNA, and that RNA is put into the machine, RT-PCR machine, and the rest is done by the machine. When you break the virus, it's not much dangerous. The RNA is not that much dangerous. Of course, we have to take care. Level 2 or 3 will be fine, I think. So same thing for sequencing also. What you need is just to lyse the virus, isolate the RNA, as you are doing for the RT-PCR testing. Then that RNA just put in the sequencing machine. And what about the sequencer in Manipur? I think there is one. I don't want to tell where is it, but there is one. It was installed recently, but I don't know whether they have the resource or not. And again, we need some permission and fund. If permission is given, some funds are given, we can sequence. We can give to the company, sequencing company. We'll isolate the RNA, send the RNA, and they will sequence and send back to us. Then we'll analyze. Sequence that machine will do only, it will give only the sequence, but we need to analyze using bioinformatic tools. That is the most important. In my life, I have done HIV sequencing and published 3-4 papers. We have done that. HIV virus is also a very dangerous RNA virus. Okay. Hello, everybody. Hello.
Yes, sir. Yes, sir.
Okay.
Yeah.
Everybody
It's the
keep
sir.
quiet. So I'm wondering where all everybody has gone. Anyway,
Yeah, we are listening, sir.
now okay, good. So now we'll come to the vaccine, the next topic, the vaccine. And you know, our body, nature has given us a protection. It's just like a shield. The immune system, the nature has given for protection, a very, very strong and very sophisticated system to defend from many pathogens, including say virus, bacteria, all kind of microbes. We are all the time exposed to the pathogen. Our environment is full of pathogen. Because of our very sophisticated immune system in our body that is given by nature, we don't get easily infection. So when a virus attack, our immune system will defend it and virus disappear. So this, I will not go into the immune system just to understand that antibody and the antigen, that part, just to come that I'm showing that our immune system. So our immune system there is a very complex system. Even our skin is also a part of the immune system. Then in the bone marrow, we have the white blood cells, the thymus, the spleen, and even in our respiratory system, from the nose, starting from the nose and the lungs, all the lining, they are covered with some kind of hair-like structure and mucus. So that are to protect us or our body from this microorganism, the pathogen. So little more detail is that this our immune system can be divided into two. One is innate. Innate is the physical barrier. For example, when we when our something enter into our eyes, then the tear comes out. That is the defense system. And our skin protects us from infection. When we have a cut in our skin and we get infection, then so we apply, when there is a cut in our skin, then we apply antibiotic and the protective medicine. And adaptive is some kind of cellular. It involves specialized cell in the body. Yeah, this also there's two types: non-specific and specific. Non-specific, some neutrophil and a macrophage. These are important. Then in the specific defense system, immune system is very now is interesting. So all the vaccine and everything will come to this specific because they have a very sophisticated mechanism of defense. This specific we'll discuss. So when there is a infection, blue color round, they are, let's consider they are our B cell in our body. And this this color is a virus. They suppose it's a virus. In the immunological term, we can call it antigen. Any foreign particle which can stimulate antibody production in the body is known as antigen. So we can consider, we can call it antigen also, or the virus. So when this virus or antigen enter our body, they bind to that B cell will bind and try to destroy it. Our immune system, this antibody involving immune system, they can recognize this is the pathogen, this is the virus, this is the foreign coming to the body, and this is our own protein, our own part. That's the beauty of this our immune system. So once this antigen, which is a foreign particle, enter our body, that B cell will be activated, and they will synthesize antibody, and this antibody will be released in the blood, in the plasma. And this antibody tried to destroy, neutralize this antigen, this virus, this foreign particle. And some of the cell, some of the B cell, they remain in body as a kind of memory. So one antigen and one antibody is very specific, just like key and lock. I will show you in next explain. Let me explain it in the next slide. So now we know that some of the B cell, they remain as a memory B cell. So if you look detail in the spike protein, the antibody will not recognize or bind to the whole spike protein. It will bind to some small part. So for example, this part has a protein structure that have small different cell. And the antibody will be raised against this one, will have this particular cell that easily fit, just like log and key. And this one particularly is a pawn-like antigen structure, and this antibody will have the V-cell structure. So easily it binds. So antigen and antibody, they are very specific. So suppose these are the variant. I just make it a bit simple. A, B, C, D, E. Suppose let's consider these are the antigen, or these are the variant COVID-19 virus. So, for example, a variant A has entered to the body and raised to stimulate the production or trigger the immune system, activate the B cell, and the antibody are synthesized in the body. Then this antibody, particular antibody, will recognize only this antigen. and destroy it. But when another variant comes which has slightly different structure, this antibody will not recognize it. So what is vaccine? And how vaccine are made? So this, so let's little bit try to understand. This is a active virus, and this active virus can be inactivated. A virus has two parts, mainly two parts. This virus structure can be divided into two parts. One part is to act as an antigen to activate and B cell and produce an antibody against it. And another part is make the people sick. This infectious. It becomes infectious. So if this live virus means active virus, some part which is going to cause the infection is modified or inactivated, or only a part of the virus which is strongly induced antibody production synthesis in the body, that can be used. So now this inactivated modified or part of the virus, it acts as an antigen and it makes antibody in the body. This inactivated modified or part of the body is carefully designed, will not cause infection, but it will produce antibody in the body. So when next time the same virus come, when this next time, what I mean next time means after the vaccine injection, if the active virus come, now our body is ready. Our body is ready with the antibody to destroy it. So within no time, in a very short time, the virus will be neutralized or destroyed because antibody are already in our blood. This is the idea of vaccination. Vaccine designing can be done with three approaches. One is the whole virus, for example, approach. That is inactivated, as I mentioned earlier, and attenuated means modified. And the viral vector vaccine, it is somewhat complicated. It involves it needs some understanding of molecular biology. So another virus is used to make the vaccine of another virus. So it's a little bit complex. So let's skip this one. But let's say that the one approach is using the whole microbe approach. Another is a subunit approach. For example, that S protein, the spike protein, is the main one. Is the main one who is going to act as antigen. So this subunit can be synthesized or purified and used as a vaccine. And another very new technology is the genetic approach. It used the genetic of the virus. It's a very new technology. It was not done before. I will come later on. So let's see the advantages, the advantage of this different approach of vaccine manufacture. So an activated vaccine means the whole virus is inactivated with the help of some heat or some kind of radiation or some kind of thing that it is destroyed, but it is still the whole virus. It's easy to prepare and safer and produce a lot of antibody, which easily neutralize the virus. But it has a small limitation, like it is not good for elderly people or immunosuppressed patient. Means, for example, cancer patient. In the cancer patient, because of the anti-cancer drug, their immune system is weak. So if you give this kind of vaccine to these cancer people, then that is going to be good for the patient. And little bit attenuated, it's a long process. So it needs, it involves a lot of some toxic chemical. So so usually it is not used often. Then subunit, I already mentioned it's a high cost and lower antibody, and it requires adjuvant, means another complex molecule. And the viral vector, it needs a lot of effort to identify which types, which virus, which type of virus, because this vaccine, for example, say COVID-19 vaccine, to put into our body, they're going to use another vaccine. Of course, that vaccine is not going to be harmful. And DNA vaccine and RNA vaccine, it was not currently licensed to use in human. Same thing with the RNA also, it was not licensed to use human. But now, DNA vaccine is under trial. And again, RNA vaccine is now approved for emergency use authority. So Pfizer and Moderator, this type of vaccine, they use RNA vaccine. It is the first time to use this genetic material, RNA, especially RNA vaccine, is the first time during this COVID. It's not yet used before. It was under trial for the cancer patient. And when the COVID come, and they just try give the authorization for emergency use. So there are many vaccines that are approved by WHO for emergency use authority. Means, I will explain in next slide what is the emergency use. So these vaccines are approved by WHO. And one of these is the AstraZeneca, is a viral vector COVID in India. Is a with collaboration with the export University in the UK. Then another Sputnik 3 is arriving in India very soon, probably next month. There are around 15 vaccines that are approved by WHO for emergency use. And out of that, Covaxin is not in the list, is not yet approved by WHO for emergency use. And Bharat Biotech applied, they have sent all the document demanded by the WHO for its approval. So maybe within a week or two, it may be approved for emergency use. But till today, it's not yet approved. So these are this again, and all this virus. All this vaccine, they need 2 doses, except one, which is Johnson and Johnson. It needs only 1 dose, and their storage condition. This Pfizer, they need -80 or -60. Because of that, it is not very useful in a country like our country, who has no -20 or -80, -60 storage. Without the -80, even -20 also, we don't have. And so these are the vaccine. What is emergency use authorization? A mechanism to facilitate the ability and use of medical items like drug, vaccine during a public health emergency, allowing for using of unproven medical products. Unproven. Okay, that means study is not completed or unapproved. If there is no alternative, when there's no other drug, when there is no alternative for that disease, some drug or some vaccine can be used, can be give a permission for emergency use by WHO? World Health Organization. So now during this COVID, there is definitely no any vaccine, specific medicine to control this pandemic. So some vaccines and medicine are allowed to use, even study is not yet completed. Study to prove that this medicine, this vaccine is set to use for human being, or even it is really beneficial to the patient. Even without that proof also, it is allowed to use under the emergency use authorization. It's because vaccine development it's a long process. It takes around 10 to 15 years. For example, say the first step is the preclinical clinical stage, where in the laboratory you check the molecules, the explanation plan, everything. Then you come to the clinical study with human subject. There are 3 phases: phase 1, phase 2, phase 3. Only after this completely finish the phase 3, when it's proved that it's safe for the human, it is beneficial for the patient, only that approval and the license are given. And even after the approval and license is given, the 4th trial is there. The fourth phase trial is there. Even it is given to the market, I mean to the people, it is open to the market. Still, the study should be going on. And once there is anything that side effect comes and on, that license will be cancelled. This is the normal procedure. But now in this COVID, where is the time to do all the phase 1, phase 2, phase 3? It might have done, but not in normal way. So now this vaccine, what I mentioned, that 15 vaccine that WHO has approved for emergency use, that are given to human being before approval and licensing in the name of emergency use authorization. So do you feel we are red, laboratory red, like this? I feel yes. Maybe we are red, laboratory red. But there's no option. What option? As I just now said, there's no other alternative, no option. If we don't use this vaccine, who are still undergoing, we don't use, all will die. But is there risk in future? Maybe, I think, especially the vaccine using DNA and RNA. They may have. It's my belief, but I don't know. Okay, let's talk about the vaccine first phase, second phase. There is no extension of the second phase, all these things. And this extension of second phase, we can accept only when there's a study. So let's discuss about this. I think you remember that when a B cell is activated, some memory cells are there, are left. So this memory cell will immediately react and multiply and make a lot of antibody. And immediately, when the virus comes, it will destroy it. So another thing is that, as I explained, that the different variant, when a variant 1 comes, it is specific for the variant 1. Antibody will recognize only the variant 1. When other variant come to our body, that antibody will not recognize the second one. So this graph shows the time trickle of viral injection, vaccine injection, and the level of immunity, the level of antibody in our blood. So when the first dose is given, it raises a little bit the antibody. And after some days or some week, it decline, the level decline. But when a second dose is given, immediately it suddenly increases. Suddenly increase the antibody level in our blood. And again, after some days, it declines. And when we give another dose, maybe then it increases again. So when this virus, vaccine is given here, the second dose is given here. When the virus comes somewhere here, the antibody level is already high in the body. So that antibody will protect, destroy the virus. So how much is level increased and how long this antibody level in the blood it lasts, how many days, how many months, is all depend on the vaccine, type of the vaccine. Some vaccine, as I mentioned earlier, that Johnson & Johnson, they need only one dose. They don't need the second dose. Only in one dose, the antibody is very high. When in other vaccine, it needs two vaccines. So between the first dose, second dose, after how many days gap, it depends on the type of the vaccine. Okay. Another thing is that now there is a fear in the people that suppose I take the vaccine when a particular variant is prevalent. After some time, another variant is rising. So I make an infection. Although I am protected from the first variant, may not be from the second variant. That's why some people after vaccination, they get the infection. So now a lot of studies going on in this regard. How many days will be given? After how many days? After how many weeks? Second dose should be given. That research is going on. And mixture of antibody. At the same time, give a mixture of antibody that will protect from number of different variants. Or some research is going on. A particular vaccine is given as a first dose, and second dose is given in a different vaccine. So let's see some of the research. This is the COVID that was studied in US, involving around 600 people or something like that. In their finding, it shows that this COVID before 6 week was given in between first or second dose. Now what they're saying that if they have proved, they have done the research that the interval between the first or the second day is a 12 week or more. There is a twofold higher the antibody response. So the research shows that when the gap between the first and the second dose of the COVID is increased, there's a more chance of protection from the infection. Our immunity is high. This is the finding. There are other vaccine which are under trial. Some are in process. And interestingly, one nasal vaccine spray is in process. They are saying that it's the game changer. Let's see how games will change. So another research has done where the mixed match between the vaccine. In this one, the AstraZeneca and the Pfizer is used. In some group of people, they have injected the first and the second dose, the same vaccine, AstraZeneca, first dose, second dose, AstraZeneca. In a group of people, first dose, AstraZeneca, and Pfizer in second dose. In another group, Pfizer and Pfizer. And the fourth group, Pfizer, first dose, second, AstraZeneca. When they study this, they saw that the headache is increased when this mixture of alternative vaccine messing is given. The headache, the side effect, the headache is increased when they used the different vaccine compared to when they used the same vaccine. And same thing, feverness, more fever is there when they use the mixture compared to the same vaccine. And similarly, people become more fatigue, tiredness when they use the mixture. But the immunity is higher. They have strong immunity. The people who receive the mixture of different vaccine, they gain strong immunity. So headache, fever, a little bit side effect. If they are not suppressed after 2 or 3 days, they disappear. But the most important thing is the last point is it gives stronger immunity. So I think it's better. So I think let's finish this thing. And the second is I'll come to the testing. Testing for SARS-CoV-2. COVID testing is required to detect the presence of this virus in our body. And suppose if this virus can be seen under a simple microscope, under a microscope, it will be much easier. Just we can take the sample, the throat swab sample or whatever, just put it into under the microscope, and we can see whether virus is or not there. But unfortunately, virus cannot be seen under the microscope. That's a big problem. It can be seen under the electron microscope, but it's not suitable for detection, for diagnosis, because electron microscope, you need to prepare the sample. Only when there's a virus and you enrich it and you treat this with something kind of anything, then you can see under the electron microscope. So it's not possible. So we have to use part of the virus for detection. What part? As I already mentioned, that nucleic acid, in this case, RNA. For this virus is RNA, not the DNA, of course. Then protein, that the protein S can be used for detection. If we somehow can attach some other molecule and that molecule is amplified or colored, then we can see it. So this is the virus. First thing is that the first nucleic acid, they use the nucleic acid. And the second thing is protein like this. So for the nucleic acid, the test is RT-PCR that target the RNA, or TrueNAT, or CB-NAT, which target the RNA, can be used. For antigen, S protein, antigen, repeat antigen test, ART, they used the spike protein. So RT-PCR, they are, hello?
Yes, yes, we are hearing.
Okay, thank you. RT-PCR, it involves a lot of steps. And it needs trained people. First step is the sample collection. Sample is collected from the nose swab or the throat swab or whatever. Then is put into a tube and put some solution, and you break the virus. It's known as lysis. You lyse the virus. When you lyse the virus, the RNA will come out. The protein envelope will be of the virus will be broken down into pieces, and the RNA will be released. So it's in the solution. Then to remove, we have to remove the protein or other lipid and everything, and only the RNA should be isolated. Very pure RNA should be isolated. Handling RNA is different. Handling RNA is not same as the handling protein or DNA. In our environment, there's a lot of other chemical or other stuff that destroy the RNA. So it has to be protected from these molecules which destroy RNA. Even in our fingertips, whatever some fatty, some chemical in our from our sweat or whatever, it touches the RNA, RNA will be destroyed. So a very careful steps to be done, extra care. Then you have the RNA. This RNA, you have to convert into the DNA using an enzyme for reverse transcriptase. I mentioned earlier also. So the RNA, the DNA converted from RNA are known as cDNA or complementary DNA. So now we have, instead of RNA, now we have DNA. The virus RNA is converted into DNA. The reason behind it, why we need to convert the DNA. Now, we set up a mixture using this cDNA as a template, as a target. So we put into the reaction mixture some component, the building block of DNA, like T, vitamin C. Then again, some of the molecule that will bind to the DNA and emit light. So we have added T, C, A, G. These are the building block of the DNA. And primer, we have to add primers, around 3 sets of primer, depending on how many gene we want to target. And also we have to include a internal control, primer for the internal control, and an enzyme known as DNA polymerase is added together. Then they are put in a they are put in a 96 plate for different sample. So this plate, 94 plate, you can test 96 sample, but with negative control, positive control, say minus plus, around 90, say 93 sample can be done in one go. Hello. Kariaba. I will finish soon. Let me finish. I think I have okay, almost finished. Only 1 or 2 slides. Just bear me. Then it will be put into the artificial machine. And now in the machine, artificial machine, it will be amplified. And how it is amplified is artificial real-time polymerase chain reaction. It's the reaction. The reaction is repeated for many cycles. One reaction is cycle, then is repeated for about, say, 36, 37 times. So in this amplification by artificial, a single DNA piece, after, say, 35th cycle, there will be 68 billion copy from one single copy. So it is amplified a lot. Anyway, I'll try to finish up, and this is the how analyze it, and the dye. I mentioned that some dye will be added to give the signal, to give color. They are detected from tube. And the case, just let me. Hello, can I continue a little bit? Can I?
Yes, sir.
Okay. And this is the ART test, the antigen test. Antigen is very simple. It's very simple. Like RT-PCR, it involved a trained person to handle it, and there are many steps. There are many steps. There are steps which, if the person is not trained, it can get wrong. Positive become negative, negative become positive. Okay, but in case of ART, it's very simple. It's just like so women do the pregnancy test. It's just like that. Just put the sample here, and it just the signal will come up. The line will come up. Okay, so there is a disadvantage. Say this ART test is based on the antigen antibody interaction. So antigen is the virus. Antibody is the antibody against the virus. Okay. So as I said that before, they have form a complex. When they form a complex, the complex become insoluble. Okay. So and if you add some molecules, attach some molecules to the antibody, then they become insoluble complex and give color. Okay, and when there is no, so then it's positive. When there's no antigen in the sample, the person is there's no virus, then the result will be negative because it's so complex will not be there. But in case the antigen is less, means the viral load in the patient is low. The patient has the patient is infected, but the virus number is not much. Then when you do the test, less antigen, no complaints. Then what is that? Then is a false negative. That's why. That is the reason why if in ART, if the result is positive, it is almost 100% positive. If it is negative, we have to confirm by other tests. Because it may be, when it's negative, it may be false negative. Because antigen is there, but that antigen is not enough to make complex with the antibody. So this is the plan that have been done. If this is positive, if artificial is positive. If test negative, artificial is negative. If test positive in ART is positive. If test negative in ART, then there may be false negative. Then it should be confirmed by artificial. Should be confirmed by artificial. But whatever it's doing or not, that's another question. But when I look the recent the notice they have every evening they release from the government site, when I look there in Manipur right now, RT test is among the older tests. They in Manipur, RT-PCR is used, TrueNAT is used, CDNAT is used, ART are used. Out of this, ART from 45% to 60% they used. TrueNAT and CDNAT only 7 to 9%, and artificial 35 to 40%. This is not good. If the people who shows negative in the ART is confirmed by artificial, that is good. But if we don't confirm, just ART negative, then you go home. And if it is a false negative at home, he's going to infect other people. So I think why this number is increasing, the positive number increasing, is because of this problem. So it's my suggestion to the authority that to set up RT-PCR testing facility center in all the districts of Manipur. It is very much possible. Some people said there is typical no electricity or not. In this 21st century, why not electricity? It can be arranged. Even if electricity is not there, then generator can be arranged. There are many things can be done. It's the 21st century. And the result of the RTR should be given within 24 hours. Many people get ART-PCR result after 4 or 5 days. Before knowing the result, they already spread the disease. Only when the result comes, he feels that he is negative, he is positive. That time only he take care. Before the result comes, he mix with other people, then spread. So result of the RT-PCR should be given within 24 hours. And the quality control should be done for the RT-PCR weekly. Because there may be cross-contamination. The center, the room should be fumigated or clean every weekly. And to conduct a mass test, there's a different way to do mass test. Some people are saying that it's not possible, not possible. If we want, it's possible. That's why to do the mass testing, if you do with the ART and the negative ART result are not confined the RT-PCR, is useless. You have, when you do the mass testing, first thing is you have to set up more center for the RT-PCR. Most of the tests should be done by RT-PCR. It's the gold standard. If everybody knows that our ART is not accurate. It's very doubtful, that especially when it's negative. So when we do the mass testing, we should keep in this in our mind that RT-PCR should be used maximum. And those positive should be isolated. When we isolate it, we have to isolate deliberately, not for just to say we have done it. Deliberately, what mechanism? It may be home isolation or quarantine center, whatever. But when you do that home isolation, you have to do perfectly. You have to make it sure that the person will not spread in his family or her family. Then some other people can be isolated in quarantine center, but deliberately, so that the virus not spread. Just not just to announce in TV. And mass vaccination must be done at the earliest. There are only two vaccine types. They're available in India. We should grab the vaccine available in other countries somehow. If we cannot, the Indian virus cannot be produced in a short time, then we need the vaccine that available from other country. Thank you.
Thank you, sir.
Thank you so much.
Thank you very much. Any question?
Professor Anju has given very nicely with well-designed presentations about the theme of the topic, that science of variants, vaccine testing and their management. Right from the very inception of the emergence of COVID-19 way back in December 2019, crores of people have been affected all over the world, and India is also not an exception to that. More than 170 millions of people have been affected. 3.5 million have died. And coming down to the context of India, more than 2.8 crore people have been affected. More than 3 lakh people have also died. And we are trying so hard in order to control and prevent from this very dangerous and disastrous pandemic. In the meantime, we have been witnessing emergence of so many mutations, emergence of different variants of COVID-19. At the same time, development of different types of vaccines also we are witnessing. And we are trying hard all over the world to control from this dreaded disease. And the way how management is taking place in different parts of the world is very important issue now. So in today's series, organized by Fit India Campaign Committee, Manipur University, and Fit India Club, Manipur University, our invited speaker, Professor Shanjukumar, has nicely and very comprehensively delivered on the topic chosen. He has given a very well-designed presentation touching upon all the facets of the variants, vaccines, and management perspectives of COVID-19. It was very impressive, very interactive, very informative. Now we have certain questions from the participants. We have around 5-6 questions from 4 participants particularly. Now I will take up these questions one by one, and our invited speaker is requested to kindly give answers to those questions. Now let me take up one question raised by Pongrai Vidyarani. She rose 2 questions, and the first question reads, if the variant is shown to be more resistant to vaccines, that would seem to be a problem. What can be done about it? And the second question is all about, can we mix and match between the vaccines? As for example, the first dose with Covaxin and second dose followed by Covishield. So in this regard, I would like to request our invited speaker to kindly answer to these 2 questions first, raised by Pongrai Vidyarani.
Okay, thank you.
Yes, over to Professor Sanju.
Yeah, thank you. I will answer the second question first, whether we can mix the Covishield and the Covishield, right? Okay.
Yes.
I don't know about this one because we have not done, in Indian research is not done. But I mentioned the Pfizer and AstraZeneca, they have done the research. So, but most probably it will be better, like the 2 businesses I've mentioned. In India also can be studied, in India also can be studied, do the 2 vaccines available in India. But without a study, it's not good. We should not try. I don't encourage to try ourselves. And another thing is that I heard some doctor in India, they are taking the third dose. It's not officially, but privately, they are taking the third dose because they are exposed too much to the virus. So in the second dose, after say few weeks, say 5-6 weeks, second dose, that our antibody level may be declined. But for people, normal people like it will be okay. But the doctor who are always handling positive, they are trying the second dose. And it's not only the two vaccine I mentioned that they try mixed. Recently, I found in other vaccine also they're doing combined vaccination. So research is going on. So unfortunately, in our India, this kind of research is not doing. And the first question, what is the first question?
It is about if the variant is shown to be more resistant to vaccine, that would seem to be a problem. What can be done about it?
Yes, that's the worrisome. Most of people are worried about that. If the new variant is not effective to the antibody race against the variant 1. So I mentioned the paper. The mentioned that after second dose in other country they are trying third dose as a third dose with different antibody. Sorry, with different vaccine. So different vaccine will have different antigenic property, and so they will raise different antibody. So only option is that when there is a new variant and it is resistant to the first vaccine, then we have to look for a new vaccine. Okay, next question, please.
Okay, Congratulations, Vidyarani is quite satisfied with the answers. Now, let me take up another question from Ronald Singh. The question is, what is the possible way to find out the common antigen in coronavirus? Yes, this is the question, sir. What is the possible way to find out common antigen in coronavirus?
It will be very difficult when you talk about the common, means it will be effective to all the variant. Whatever variant comes will be effective. In the slide I mentioned that the vaccine used the whole cell, inactivated whole virus, not cells, whole virus or attenuated whole virus will be more effective because it will have more part of the virus. So when it injected in the body, a different variety of antibody will be produced. But as you know, virus mutate when there is a pressure. Even now, HIV virus cannot be produced because HIV keep on changing its shape, its structure. So because of that, HIV virus cannot be effective virus cannot be produced. Similarly, in this case also same. Will be hurt. Only thing is that different type of virus, different type of vaccine should be started immediately. And they are also trying, as I mentioned, they are also trying around 7-8 different vaccine, and whole world also trying different vaccines. So luckily, it may solve that when any new virus comes, one or two of the vaccine which are undergoing development may help in future.
Thank you, sir.
Okay.
That means we need to go further for further research and all. Last but not the least, I have one more question from Dr. Ranjan Khetrimayum. His questions is all about production of better region between RAT and RT-PCR. So his question is about, does it true that RAT gives better result than RT-PCR?
Okay.
Yes, sir.
Can I answer? Okay.
Yes, sir. Yes, please.
I don't know what do you mean by better result. It's not the sensitivity of the test. RT-PCR, it target the RNA and it amplify even a single virus RNA is found in the sample, it will be amplified through number of cycles. Single RNA sample will be amplified up to 68 million copy. If you run the cycle up to 35. Okay, so it's very sensitive. What I mean is very sensitive. It amplifies and detect it. But in the ART, this protein is not amplified. It cannot amplify protein. So whatever is there in the sample, that you have to measure. So when you measure, it is small quality, you may not detect. We are using for detection in the ART some compound attached to the antibody, which is color. Okay, so if the number of virus is less, the color will be very less, and in our human eye may not detect. So it will become negative. That in the PCR is amplified. Moreover, the signal is detected with a detector using laser. So it's very sensitive. So you cannot. Only thing is that RT-PCR, it takes time. RT-PCR, it takes time. ART it takes, say, less than 30 minutes. Okay, just you put the sample, wait for some time, then run. It's just like the when some female do the pregnancy test. That much easy, but the sensitivity is very less. I
Okay,
think.
sir. Yeah, that
enough
is quite satisfied. Yeah, quite enough, quite enough. And the participant is also quite satisfied with that. And now one private question from Sanam Thabam Rudrajit again. The question is, sir, there is many patients doing home isolation. Is there any possibilities the virus get transferred through households, waste being thrown outside? Yeah, this is very important question, I think.
Can I answer now?
Yes, sir, please.
Yeah, it is possible. It is very much possible that household item or anything that touched by a patient, COVID positive patient, it's possible that it is contaminated. And there's a many record that on surface it can stay up to hours. So when at home, if there is a patient, then we have to use sanitizer to clean door, knot, door handle, and everything. It's better to wash with alcohol or hand sanitizer.
Yes,
What about touch by hand? And those things, those stuff throwing out from the house of the COVID patient, it's better to burn it. It's the best way is to burn. I think it's okay.
Answer is very perfect, sir. So now, no more question. I actually invited to all the participants to submit their feedback as well as raise questions. So 7-8 questions we received, and answers for those questions have been already given by the expert. So right now, no more questions. So I should say once again, thank you so much, Professor Shanjukumar Singh, for your nice, very well designed and very comprehensive deliberation on the topic of the day. And you have covered almost all the facets of the topic. And all the participants are very much enlightened with your very novel ideas about different types of vaccines so far developed and mutations that have taken place about the coronavirus and the way how we can manage all these things. It was a very purposive and a very useful talk organized by Fit India Campaign Committee Manipur as well as Fit India Club Manipur University. Now it's time to say goodbye, but before that, let us have a proposal of thanking to all the participants as well as the panelists. So in order to take off now, I would like to hand over the mic to our coordinator of the series, Dr. Santosh. Over to Dr. Santosh, please.
Thank you, sir. Thank you, sir. So very brief scientific presentations on COVID-19, science of variants, vaccine, testing, and their management. Thank you so much. We have got the knowledge, everything from there. So in the conclusion parts, I, on the behalf of the Fit India Campaign Committee and Fit India Club of the Manipur University, I would like to propose a vote of thanks to today's panelists, respected Professor L. Shanjukumar Singh, Head of Departments of the Biotechnology, Manipur University, as a speaker. I thank you so much for your sharing your wonderful your expertise of your knowledge, very amazing about your information of the scientific backup of the COVID-19. Thank you so much for your sharing the knowledge in these 4 sessions of the national talk. Thank you so much. And secondly, I'd like to
Hello, can I say something?
Yes, yes.
I thank Dr. Ibohal Singh and Dr. Raj Ketan and Dr. Santosh for inviting me. and all the participants for listening this talk. I thank you all.
Thank you, sir. So secondly, I would like to propose my vote of thanks to the Dr. C.S. Ibol Singh, Associate Professor, Head Department of Library and Information Science, Manipur University, for moderators as the sessions for successfully conducting these sessions. And then I like to propose the vote of thanks to the co-moderator, Dr. C.S. Rajketans, Department of Manipuri. So I thank you so much from my bottom of my heart to the co-moderator and moderator for conducting smoothly these sessions grand success. Thank you so much. And especially I would like to thanks to all the participants, all the learned member, teaching, non-teaching staff, and the students, and Mami TV as a media partner, and viewers and listener, everyone. I thank you. Thank you so much for your motivations, for your patience, for this session's grand success. And then I request you for next episode, 2nd June 2021. We have a topic, a very good topic on stress management during the pandemic. Our speaker, we have Dr. R.K. Lenin's RIMS Department of Psychiatry. He will speak about management of stress during the pandemic. So I thanks once again all of you. So in this occasion, I like to thanks everyone and say bye-bye everyone. Thank you so much.
Okay.
Mm.